So I'd like to introduce Professor Frederique Deiss. She's an assistant professor at IUPUI School of Science. As I mentioned, analytical chemistry is an essential element behind diagnostics. And Indiana is a leader in this science, and it teams up well. And I was privileged to be teamed up with Frederique.
Thank you, Michael, for this introduction. So as I mentioned, I'm an assistant professor in the Department of Chemistry and Chemical Biology at the Indiana University, Purdue University, Indianapolis, IUPUI. And so what we are doing in my group for like the last few years is to try to develop electrochemical bioanalytical tools. And so we do that in particular using paper microfluidic spectroscopy. So what that means is like, in addition of like kind of advancing the fundamentals of chemistry, like you will typically do in a department of chemistry, we are also trying to do applied research, meaning actually having projects which will answer a societal question. So any type of challenges that have been identified and trying to specifically find a new way, or better way if possible, or alternative way to answer it. And so in particular, we do this with electrochemical analysis because, well, electrochemistry is known to be sensitive, selective, and quantitative, and allow like nice analytical detection. But so what we also do in our system is we are trying to develop tools that will be specifically for in-field and point-of-care application. And when we do that, we don't only focus on the chemistry, but we also focus on the sampling. How will we collect the sample? How will we transport the sample? How will it be stored? And all these different aspects which go around making a point-of-care devices. And so our application go to preventive care, diagnostic, environmental monitoring, and also forensic. And so today I will try to show you a few projects that we have ongoing in the lab and try to highlight some of these particular aspects in each of these examples. And so you can kind of see in the slides scattered all around multiple paper-based devices. And so I will say 2/3 of our project are actually using paper as a substrate to make low-cost, portable, versatile point-of-care devices. The first project I would like to introduce to you is something we are doing for oral preventive care. And the idea is that you can actually know if someone has chances to develop a cavities before they have a cavities by looking at the pH of the plaque on the bacteria, the saliva around the mouth. So if you're below a critical pH of 5.5, so typically you can begin to know that the bacteria responsible of cavities will be very happy, and they will also maintain this very low pH to begin to start the erosion of the enamel of the teeth and to form a cavities. And so what we want to do is we want to make a little like paper-based device like you can see here, and that's like, because they are so portable, I could bring with me in my home. So you can actually have them and be able to measure the pH at different position in the mouth to do it, but having no chemicals on it, because who would like to kind of put a bunch of like pH paper or chemicals in your mouth when you're testing it? So the idea is to actually only use graphite, which is pretty much what you have in a pencil, minus actually the glue you have in a pencil, so it's safer than a pencil, putting into contact with the teeth and measure a pH. And so to do that chemical-free, if we can say, what we are doing is we are relying on some oxo group like quinone, which are present just by reaction of graphite with the air. So if you have graphite, if it's in presence of air, you form this type of quinone group, and their redox activity is actually sensitive to pH. So depending on the pH, you will have different peak, and so you can see this type of peak here, which will kind of like shift depending on the pH, and then you can do some kind of calibration curve over multiple pH and such on. And so this is a project that we started, of course, like pretty much for 2 years in the lab doing solutions based and developing all these aspects. But then now we have moved to artificial saliva, and our next step is now to use real saliva and real bacteria biofilm responsible of cavities with our collaborator in the School of Dentistry of IU. And so another project we have, staying with the bacteria detection, is to try to have a system to detect bacteria at a very, very low limit of detection. Meaning our goal would be to really go down to 1 bacterium. because we know that even one bacterium can be lethal in some type of, in some pathogenic disease. And so very often, the current method of detection will rely on some type of amplification or culture. And as Michael mentioned before, we have kind of like 3 paradigms which we use for point of care diagnostic. And so 2 of them are like isolation and concentrating our signal. So what we are actually trying to do here is to be able to achieve this very low limit of detection. Instead of trying to increase the amount of bacteria, we're actually decreasing the volume by encapsulating our analytes into droplets. And so we are pretty much locally having a high concentration, not because you have more bacteria, but because you have a very small volume. And so we are currently developing in my lab, like all the kind of system, doing some very basic microfluidic device, like Steve Riley will show you something way fancier than what we do. We stay very basic because we are more focusing on the chemistry. But what is the impact, especially of this project, is we are doing what we call the reverse assay, which means that if we have no bacteria in our sample, so bacteria is this little pink thing. So if you have no bacteria in your sample, you will actually have light emitted. So you have a signal. But if the bacteria is present, you have no signal. And you might wonder, why can this be relevant when you're trying to do a low limit of detection? Well, this is all good when you're doing detection in solution that you have created. But if you go back to a real sample, you will have a lot of other species in what we call the matrix of a sample. So the other element present in sample, but what you're interested in. And these other elements might actually react. They might actually artificially increase your signal, creating false positive. They might decrease your signal, creating false negative, and such on. And so having a reverse system like this for this project actually allow us to have 90% of our droplets, which does not have a bacteria in it, to give us a baseline, which will take into account all these other elements present in your complex sample. And so this would also allow us to do it with a known sample, if we can call it, like a blood sample, when we typically know the interference, but also way more complex sample, like having food, beverages, soil, where you will have almost unknown interference that can mess up your reaction. And so having 90% of your signal, which is actually coming from all this interference, give you a way better baseline that if you're trying to do a standard before or do another reference before your system. So this is actually one of our current ongoing project, really focusing in being able to analyze complex samples when we don't know what is in it. And then I'm like now changing gear as an electrochemist. We like to detect ions. You know, ions, like all our redox chemistry kind of is coming from an ionic metal plus electron give you back the metal and such on. So it's kind of the bread and butter of electrochemist. And I'm sure a lot of people must have used ion selective electrode in a lot of different type of labs, which typically use potentiometry. We are actually developing new techniques which will use voltammetry and allow us to use our paper-based system in like a simplified probing system, which doesn't require to have a fragile probe and such on. And we started this project developing it for blood analysis, because actually your potassium level can help you assess the overall health of someone. It can also be very useful for disease like hypokalemia, where people cannot fix potassium and literally require their blood to be tested every week, every other week, kind of like diabetic patient, but for potassium. So trying to have a simple system like the glucose meter, but for potassium. Because it's a disease which doesn't touch as many people as diabetes, well, you don't have currently a disposable portable system to do it at home. So people need to get measured in a lab every week or so. But when we developed this project, what was very interesting for me is to realize that I had people coming from all different areas of science and telling me, oh, this could be very useful for what we work on. So for example, a collaborator in the School of Medicine, Department of Pathology, for the forensic aspect told me that measuring the potassium level in the vitreous humor, the liquid behind the eye, can actually be useful to estimate the time of death. And so he actually has many hundreds of samples ready. So when our test is ready, we have like liquid of dead people, so we can measure some real sample for this. People are beginning to tell me how this is so useful for water quality. That actually people, when they, in industry, when they do bottled water, they do test for potassium to have an overall idea of the of the quality of their water. It's also very important for agriculture and soil analysis because it can give you information on the overall assessment of the soil at a particular environment. So having a portable device to do it, which would be even degradable with paper, could be very useful. And then it actually circled back to the food, because potassium is one of the micronutrients or electrolytes that you can actually ingest to kind of balance your nutrition. So you actually need to know how much you have in food to know how much food you should eat to kind of bring your potassium level back to where it should be. So what I wanted to show you with this particular example is that if in our lab in chemistry we start with one particular project and we have a certain like characteristic, we are developing it for blood, certain range of concentration, we can after adapt it to many other applications in different fields. But for this, we need to actually have the collaborator who can let us know that this will allow us to, sorry, I read a question at the same time. We need to actually have the collaborator to let us know where the need is. And so, this is actually very important. And I think this is also what happened here with COVID-19. We see so many researchers, they started their work, maybe they were working on coronavirus, another one, or they were working on point of care diagnostic of virus, and they were able to shift and adapt very quickly by talking to the right people and knowing the new characteristic to transform maybe a device done 90% for something else to something which now could be useful for COVID-19. And this is so important in our research system. And so now another project I would like to go to is kind of on the other side of the field, and not so much for the diagnostic for the health of patients, but actually for forensic applications. So we have currently some federal funding for the National Institute of Justice to be able to detect an oxidizer used in IEDs, which is chlorate. So fertilizer was used a lot before, but now fertilizer is so controlled that bad people kind of shifted a lot to chlorate. Sometime up to 80% of bombs, for example, in Afghan's war were actually done with chlorate. And so we are trying to have a system which will allow you to screen quickly in the field, the system. But here again, we see the whole importance to know what kind of sample you will want to make your device for. Because you see here with the collaboration of the Indiana State Police and the Good Pastor Lab in IUPUI for the forensic program, we were able to explode our own bombs. So the police did that. But so we actually had like a pipe bomb, for example, made of metal here. You would need in such a sample to be able to swipe your device against such a metal to be able to test it. But we also made some bomb with plastic pipes. And I can show a picture for you because this was pulverized, which actually means that like all the plastic is in small pieces, which are pretty much mixed with all the little stones of the quarry we did our explosion. So that means that for this type of sample, we actually collected soil, which has a little piece of plastic. And so we need our device to be able to collect sample in multiple type of systems. So our solution when we do it in lab or after swiping system or like collecting powder. So for example, with our paper-based device, we had to come up with solution to see and test to see if we impregnate our device with certain volume of liquid, how will we be able to swipe the powder and then being able to see our electrochemical signal from that. And I mentioned earlier, so storage and transport can also be important. It's particularly important for forensic because let's be clear, my device can be useful for screening, but they will take a long time that they can be used in court and you can actually convict someone of us saying, oh yes, we think you made a bomb here. And so here's the idea is we need to be able to collect our sample, do a quick screening in field, and then bring it to a state lab for confirmatory test. And so here's the idea was to actually use similar type of system like tape and paper and encase our whole system in something which will allow it to be fully closed, store our sample, transport it to the state lab and house there the ion detection that are like the gold standard for like the chloride detection, and these have all the proof, both the screening and the confirmatory proof that can be used there further. And so this actually brings us to the project that Michael already presented a little bit to you, that we collaborated on. So we presented you some of the aspects of like the engineering behind, and I will talk to you a bit more about the chemistry that we did here, and also how relevant it was to understand how we can mix multiple devices. So as we said here for this particular project, we're trying to use a Sierra technology and being able to detect some bacteria without any culture, and being able to, in particular, identify if it would be a gram positive, gram negative, and then being also able to get right away the DNA RNA to then know if we have any type of like, to what will be the responsibility for the antimicrobial resistance genes. So what we want to do here, if you think about it, is an electrochemical assay, a mass spectrometric assay, and in the same time, we want to collect this in a membrane to do microfluidics to kind of have all the sampling happening, and then lyse our bacterial cell, which was detected, and be able to collect the DNA. So this is actually multiple steps with multiple type of reagents, different type of volume, different type of sensing device, and we wanted to mix all this. Why do we want to mix all this? Well, because electrochemistry in this case can give us very quickly a screening test to just know, is this bacteria present? And just by detecting this with an enzymatic reaction, so kind of like an ELISA type, but done by electrochemistry, we can detect the formation of a product and know that like the bacteria was there. Then the mass spectrometry will kind of give us some type of barcoding, and using a realizable mass label agent, as Michael described before, we'll be able to actually know more about what bacteria was present as well, and have like already a confirmatory test about what's going on. Might take a little bit more time our screening, but it's still very, very fast. As you mentioned before, we can do 28,000 samples per day. But then after that, you can actually do the DNA RNA isolation. And what is important here is doing the test on DNA RNA is way more time consuming and needs more trained personnel. Whereas here, we'll only do it for the sample which are already positive. So this means that you're really helping the whole backlog of tests. And so when you begin to have a lot of people sick in the same time, as we are seeing now, having this kind of like cascade of a very fast screening test, a confirmatory test pretty fast, and then a test which gives you all the information of the pathogen change and like the mutation, will actually really give you a lot of information without overusing your personnel. So this can be actually very important. And so here we needed a very, very big team to be able to develop all this project. So all the collaboration Michael already described before, and especially because we needed to make sure all our reagents are compatible. And so for this, we needed to hear from everybody, from the microfluidics point of view, for the chemist point of view, the mass label expert, the people from bacteria expert, to have all the experts together to know how we can make this work. And so today we do not have with us some collaborators, but I wanted to really highlight in particular Dr. Deiss from the Department of Pathology and Laboratory Medicine, with a key opinion leader for our industry in serving especially microbiology clinical laboratory, and who were able to give us some urine samples that was the key to make this project from a proof of concept to a real device. And also Dr. Sen and Dr. Roy in the Department of Pathology and Laboratory Medicine as well were key opinion leader for all the biofilm forming, and was very, very useful for us to make sure we can get this isolation and testing of our bacteria. And so I would like to finish on the next slide to thank all my students, because I can tell you that if the professor talking, all the students are always the one who do most of the work. And so in particular, I want to highlight Regina and Monica Baron, who are two undergraduates, were in between my lab and Dr. Max Puggia team for like the last 3 years. They are now moved to other endeavor, doing a PhD for one and a MD for the other one in IU School of Medicine. And also Dr. T.H. Bose, who is a postdoctoral fellow in IBRI, and also helped a lot my group. So we were really, these three people was a bridge between Michael's team and my team to be able to bring all our skills together, because I knew a lot about the electrochemistry, but we needed all the information about the urine samples, the microfluidics, the mass label to make this work. And so this was very, very important to be a successful project here. Thank you to all of you for listening today.
So Frederique, I know from your talk here that you really get the value of having the end user direction, and whether that's clinical or other, and of using real world samples. But what I find confusing often when I do these programs is which end user to listen to, and it just, do you have any advice for designers?
So yes, that's actually a very good point. And you saw, there was one project I mentioned, we could have multiple direction. I will say I definitely have a specific direction when we're developing one project with one particular team when we're doing this. And using real sample is so important, because I think every researcher, especially the one developing device, they don't want to just like do science. They want to actually help people. They want to help save lives. They want to help the environment, the communities. And to do this, you cannot do it with just a little sample we create in the lab. We need to have the real sample with all the interference that we might have. As I mentioned, everything which could create fake positive, fake negative, and such on. And to get this, you need collaborators. You need to have a network of people, and I will say being able to interact with teams like here with IBRI, this open a lot of doors for me to have other collaborators that can then be useful for other projects. Making sure to have, as I mentioned, people who are really there. Like Michael and I was meeting once a week or every other week, but like the girls and Dr. Tiajbos, they were there every day, and they were often in contact with both of us every day, and they were at meeting with all the other members of the team. And so they were able to really capture the characteristic and convey what are needed. So I think really being clear early on what will be the real sample, what will be the real target, and have a very clear characteristic set. If we want to do urine, you have another volume that if you're taking blood. If you're taking blood from a baby, you have way less volumes that you would have if someone pee. So kind of having this is very important, both for the chemistry, for the concentration sample, for the microfluidics and such on. So being very clear of your characteristic and talk to the experts, because we are only expert in our own zone, and we need the others and the collaboration to get there. And I would add that for me, being able to collaborate with a team like the Indiana Bioscience Research Institute team was very key. This project was on steroids versus all my other projects. It went way faster, even if I didn't have a PhD student on it. But because having someone with having all the industry mindset and is able to connect us with people like Jason Kulik at IIC, or like with Purdue Engineering and such on, you can actually really push the barrier, like having stuff happening faster, having the means. And I'm not just talking about money, but way more about the time and the access to experts to get stuff done. Thank you.
Thank you. Yeah, I appreciate that.